So, what are muscles? So there actually are three different kinds of muscles. There are skeletal muscles, there are smooth muscles, and there's cardiac muscles. And usually when people introduce how muscles work, they're talking about skeletal muscles, which is the muscle that you're most familiar with. So yes, when you do this, those are skeletal muscles. Skeletal muscles, well, there's small ones too. There's skeletal muscles that, skeletal muscles are under voluntary control. And that contrasts with cardiac muscles and smooth muscles, which are mostly under involuntary control. And what voluntary control means is that you can volunteer to use your skeletal muscles. Your skeletal muscles mostly, not entirely, work when your brain tells them to work. And smooth muscle also works when your brain tells them to work, but it tells them to work in an unconscious manner. You're not aware of your brain telling the smooth muscles to work. And that would be involuntary control. It's not something you can think about and make happen. But the skeletal muscles, you can think about what you want your muscles to do, and then your muscles will do that. Now there's an exception to this, and that's seen with reflex arcs. In the reflex arc, it's not under voluntary control. It happens in an involuntary manner. And this would be, for example, you accidentally touch a hot stove, or put your finger onto a pin, or step on something sharp, and suddenly your body just does something to move you away from whatever it is that's causing this pain. And that's an example of a reflex arc. We've got reflex arcs in our bodies that are not based upon your feeling pain, but what they all are basically movements of your body, reactions of your body, or responses of your body that occur in a manner that's really not under voluntary control. They just happen. When you touch your hand to a stove and it's really hot, you pull back your hand, the withdrawal reflex. What's happening is that the nerve impulses are going up your arm to your spinal cord, and then rather than going to your brain, which is where the voluntary control comes from, it just goes right back out of the spinal cord down what are known as motor neurons to the appropriate muscles that cause your arm to pull back. That's a reflex arc. And the reflex arcs are useful because they're very fast. If you have thinking going on, it means that you've got multiple neurons that are being used, and these neurons they are used in a serial manner, one after another, and it takes time to have nerve impulses propagated down a neuron, move to the next neuron, move to the next neuron, move to the next neuron, and so on and so forth. If you can reduce the number of neurons that are involved, these are nerve cells, and also if you can speed up the connections between the nerve cells, then you can have, in fact, if you can lower the distance that you're traveling along nerve cells, you can have much faster response times. And so a reflex arc, you'll have some kind of sensation, it'll go to your spinal cord, and then it'll go back to the motor neurons that innervate your muscles and make your muscles do things. But by and large, we have voluntary control over our muscles. But you still have these motor neurons. And the motor neurons still are coming from the spinal cord, but the impulse that tells these motor neurons what to do, and therefore the muscles what to do, is coming from our brains. And whatever voluntary means in our brains, whatever it is that consciousness means in our brains, it's coming from our brains and making things happen. So when we're talking about muscles, we really can focus on skeletal muscles, which is the way it's usually introduced in a physiology class, and we can start with motor neurons. So, I've got this list that I put up there, and as those of you who got in early can attest, I did this whole list, not that it's a very long one from memory, except that I couldn't remember that word there. I had to look it up. But at any rate, this is showing what happens in the course of muscle contraction, starting with the neuron, the motor neuron, that's innervating the muscle, and ending up with the muscle contraction itself. So the neuron we can draw something that looks like this. And at the end of this neuron, there are going to be these processes. So this is an axon of a neuron. And we have action potentials. Also known as depolarization. Also known as a nerve impulse. And these propagate down these neurons. So this is basically plasma membrane, and this would be cytoplasm in there. They propagate down these neurons, and they're a way of taking information from one place in the body and moving it in a very linear manner to another place in the body. And we'll go over what these nerve impulses are all about when we talk about the peripheral nervous system. But for understanding your skeletal muscles, it's important to at least have some sense of this propagation of information from one place to another. So the axon ends up in these processes, the axon termini, and with muscles and their innervation, the termini are called neuromuscular junctions. So these are junctions between neurons, the ends of the axons, and muscles. So if we take that and we kind of blow it up, let's see a different color. So this is just one of these small things here, and we'll make the muscle cell look like that. So this is the neuromuscular junction. Right there. And so you've got this action potential, and it propagates down the nerve, and it ends up at this neuromuscular junction, and at the neuromuscular junction, a chemical called acetylcholine is released. That's a neurotransmitter. It's the neurotransmitter that essentially connects the information coming from the nerve cell, the motor neuron, to the muscle cell. Now, what happens is that there are these things called vesicles inside of the nerve cell, and they fuse with the membrane, and they release this acetylcholine, and the acetylcholine diffuses across a very short distance, this gap here, the synapse as it's known, and they reach the surface of the muscle cell. Now, the surface of the muscle cell has on it, on the surface, I'm drawing little dots here to indicate it because the scale, I haven't given myself much room to draw them. But on the surface of the muscle cell are these ligand gated channels. And those are membrane proteins. And what a ligand is a small molecule that binds to something else. So here the ligand is acetylcholine. It's just a small molecule. And the gated channel is an example of a membrane protein. So this is this blue thing here. That's the surface of a muscle cell. The muscle cell is much bigger than that. But the muscle cell, like any cell, has a plasma or cytoplasmic membrane on it. And within that membrane are membrane proteins. And at these neuromuscular junctions, you have membrane proteins that are described as ligand-gated channels. So the ligand is acetylcholine. It's released by the motor neuron, at the neuromuscular junction. It diffuses across the gap here, and it reaches the ligand-gated channels, and it binds to the ligand-gated channels. So you've got information propagated down your axon, depolarization, action potential, nerve impulse. It reaches the terminus of the axon, and it stimulates the release of acetylcholine, and the acetylcholine binds to the surface of the muscle cell at these ligand-gated channels. So from this point on, the propagation of the signal that's telling your muscles to contract is going to be associated entirely with the muscle cell. Because at this point, the motor neuron's done its job. It's delivered the signal to the muscle cell. Now the ligand-gated channel, it's gated, which means it has a gate that opens and closes. When the gate's closed, nothing can pass through it. When the gate is open, then things can pass through it. And what passes through this channel are sodium and potassium ions. So Na+, that's sodium, and K+, that's potassium. But we're not going to worry about those details until we get to talking about the nerve impulses and how they work. Suffice it to say that the same action potential, depolarization, nerve impulse that occurs down the axon also takes place in association with the muscle cell. So you've got information that can move down a nerve cell. The same type of information can move down the muscle cell. And in fact, what happens is you have the depolarization, the action potential, and nerve impulse comes to the neuromuscular junction, releases acetylcholine, and that serves as the signal via these ligand-gated channels to initiate another equivalent to a nerve impulse, a depolarization event or action potential in association with the muscle cell itself. So the muscle cell is a cell, it's a long cell, it's got a plasma membrane. So now I'm going to do a blow up of the muscle cell. I'll continue to do it in blue. You're going to leave a gap there. So the muscle cell is a long cell, and what I'm showing here is simply a cross section of it, just a little section of it, where you have plasma membrane up here. And plasma membrane down there. And an action potential, once initiated at these ligand-gated channels, can propagate along the plasma membrane. And what you have is the nerve impulse, or not the nerve impulse, the depolarization event, the action potential. It moves down. And again, we'll get to how that propagation actually occurs when we talk about nerves. But it's the same kind of propagation as you see in the nerves. And it heads along in a linear manner, just like you have a linear movement of information down the axon. And it reaches these things called transverse tubules. Or T-tubules. And what the T-tubules are basically tunnels. They are continuous with the sarcoplasm, the plasma membrane, so they're basically plasma membrane. They're basically membrane. And the same kind of depolarization that moves along the membrane, the plasma membrane, can just continue along these T-tubules. And what the T-tubules do is they move this depolarization event, this information, from outside of the muscle cell to deep within the muscle cell. The muscle cells are big. They're multinucleated. They're actually formed by the fusion of multiple muscle cells. So they're big things. If you want to get information deep inside of a muscle cell quickly, which of course we want to do because we want our muscles to react, to contract, and we want them to contract, we have to deal with the problem that the muscle cells are relatively large, and we deal with this by essentially having the equivalent of like a wire that goes from the outside of the muscle cells to the inside. So we've got information that moves down the motor neuron. That information is transferred to the muscle cell. That information moves along the surface of the muscle cell. And then the information, the to contract signal, gets thrown into the interior of the muscle cell quickly via these tunnels called transverse tubules. Okay, so at this point, we're getting close to the contraction. What we have in association with the transverse tubules, and I'm not going to draw these terribly accurately, is something called the sarcoplasmic reticulum. So now we're inside of these cells. We're inside of the cytoplasm. The sarcoplasmic reticulum is an organelle that's found inside of the muscle cells. And in fact, it's an organelle that's related to the endoplasmic reticulum that you find in ordinary cells. It's a member of what's known as the endomembrane system of a muscle cell. So now we're inside of the cell. Now when we started out, I told you that we had cell biology, and cell biology is really not the thing you really emphasize much when you are studying physiology of the whole body. And you often think of the cells as black boxes where, you know, kind of information goes in and stuff happens. And I said, but there is a couple of examples where we are going to be focusing on what goes on inside of the cells. And I specifically said, and the muscle cells are where we're going to be for sure emphasizing what goes on inside of the cells. So at this point, we're now into the realm of cell biology within a muscle cell. And this is the sarcoplasmic reticulum. So the sarcoplasmic reticulum has inside of it, it serves as a storehouse for what are known as calcium ions. Or you can draw it calcium 2+. So Ca is the abbreviation for the metal calcium, and this is an ionized form of calcium. When you drink milk in order to get enough calcium in your diet, this is calcium. This is what we're talking about. Oop, I've got my little, there we go. So Ca++ or Ca2+, or Ca drawn like that, that is the metal calcium in an ionized form. It actually has, it's lost 2 electrons, so it's 2+. So this equals calcium ion. It's the soluble form of ion inside of our bodies. So the sarcoplasma reticulum serves as a storehouse of calcium. And when you have a signal that moves along the surface of a muscle cell, and then this depolarization signal moves down into these transverse tubules, the transverse tubules are found in association with the sarcoplasmic reticulum, and the signal going down the transverse tubules stimulates the sarcoplasmic reticulum to release calcium ions into the cytoplasm. So we'll call this cytoplasm. So this is actually not in the cytoplasm, and actually the interior here is not even inside the cell. It's kind of like our digestive tract, where the interior of the digestive tract, it's not the lumen, it's not in our tissues, it's actually outside of our body. It just happens to be surrounded by our body. So the T-tubules, the volume inside of them is not actually inside the cell, they're just surrounded by the cell. The sarcoplasmic reticulum is inside the cell, and it's suspended in the cytoplasm, but its interior is a different, the word is compartment, from the compartment that the cytoplasm consists of. And so what happens is that this can store these calcium ions, and they can't reach into the cytoplasm unless there is a signal received by the sarcoplasmic reticulum. It actually comes from the T-tubule, and it tells the sarcoplasmic reticulum to open up membrane proteins, another gate, in the membrane of the sarcoplasmic reticulum that causes the sarcoplasmic reticulum to leak calcium ions into the cytoplasm of the muscle cell. And this calcium ions actually are what serves as the signal immediate to the proteins that are actually involved in the contraction of the muscle cell. It serves as the signal to initiate the contraction of the muscle cells. But the signal occurs through a number of proteins, and it's also a multi-step process. I'm looking here to make sure I've gotten everything. Yeah, we're at this point. So now we have troponin. So troponin is a protein that binds to calcium. It binds reversibly to calcium. So when there's a lot of calcium around, troponin is bound to calcium. When there isn't a lot of calcium around, then troponin releases its calcium and it's not bound to calcium. Now when troponin is bound to calcium, it causes a second protein, tropomyosin, I think that's how it's spelled, which is the protein that serves to prevent the muscles from contracting. So this basically puts a brake. You think of it as the emergency brake on your car. Your car is not going to go forward unless you release that brake. So this is the emergency brake, and what releases the brake of the tropomyosin is this complex between troponin and calcium. So when calcium's not around, troponin prevents tropomyosin from being released. And when calcium is around, then the tropomyosin can be released. What does tropomyosin interact with that prevents the contraction of the muscle cells? This is a protein that you've probably heard of. We certainly mentioned it in class already, and that's actin. So there are these chains of actin, and these chains have an association with them, this tropomyosin, but when you have calcium bound to the troponin, then you have, in a sense, a separation of the tropomyosin from the actin. And when you have separation of the tropomyosin from the actin, then yet another, we haven't used green yet, oh, I guess we have. Then yet another protein called myosin I guess that must be an S, not a C. Called myosin is able to interact with actin. And it's the interaction between actin and myosin that causes the actual contraction of the muscle. And we can get into the detail of that, but I just want to go back over here. So troponin binds to calcium and causes tropomyosin to no longer block actin, so actin becomes available. It becomes available to binding to myosin. And once myosin is bound to actin, or is able to bind to actin, And so long as you have ATP around, which is energy, then you get what's known as the power stroke. And the power stroke represents myosin basically grabbing actin and pulling it. And then it's released and it grabs it and pulls it. It's released and it grabs it and pulls it. It's released and it grabs. And this grabbing and pulling slides the actin relative to the myosin. And this is actually called the sliding filament mechanism. And that sliding of actin relative to myosin pulls together neighboring actins towards each other and has the effect of shortening the muscle cell. And the shortening of the muscle cell is muscle contraction. So, from our spinal cord, often, under control of our brain, in fact under voluntary control of our brain, we have motor neurons that come out that innervate our muscles, our muscle cells, and a nerve impulse, an action potential, depolarization moves down those neurons to these neuromuscular junctions, releases acetylcholine, which then diffuses across the neuromuscular junction to these ligand-gated channels and opens them up. And this causes depolarization and a nerve impulse to go along the surface of the muscle cell, which then, that depolarization propagates into the muscle cell at the T-tubules. And so we've got this propagation of the signal down the T-tubules, and that signal stimulates the sarcoplasmic reticulum to release the calcium. The calcium's now in the cytoplasm where your actin and myosin are. It interacts with the troponin, and that causes what's known as the troponin-tropomyosin complex to free up the actin so that the myosin can bind to the actin, and then the myosin binds to the actin, and it causes this sliding filament, the movement of one next to one another, the actin relative to the myosin, and that causes a shortening of the muscle cells and therefore contraction of the muscle itself. Now the muscle cells the muscles consist of multiple muscle cells. And when we are contracting our muscles, we usually don't use every single cell. And this allows us to have a gradation in the amount of strength that our muscles give forth. Whereas we can have relatively little force exerted, or we can have more and more and more. You don't really want to use all of the muscle cells except under emergency situations, because when you're starting to use every bit of strength you have, you start to increase the potential for injury.